You know you want to sit down?
Yeah, hang on the table.
No, I don't want to do that. Many students getting things right now. Is it ready?
Let's go ahead and get started so we can stay on time.
Yep.
People are getting fed by these 6 months for the idea of closing, but it's, there are have Helen around, so borrow it if you need it.
Okay. Come sit down. I was going to give a talk about that was titled Genome Curation, or a case for genome curation. And then yesterday I changed my mind, and so it's now called Never Trust a Thank you. So hopefully, over the next few minutes, I can bring you into the mindset of the importance of genome curation and how this too can help your work. This is a continuation of Morgan's work, effectively. She has walked us through all of the data that we have available, and I would like for you to start thinking about the things that we are doing with these genomes at the level of curation. And to start with that, then I'd like to introduce many of you to the concept of biocuration. For many of you, you have heard me talk about curation before, but many of you also have never heard of this before. So just in the context of genomics, that curation is basically the collection, annotation, and validation of knowledge that is disseminated through biological databases. And it covers, sorry, you want to say something about the air conditioning or something? Yeah, sorry. Okay.
But it's so loud.
Oh, it's hard. You can't hear.
Then you can either reduce the fan speed and put a lower fan speed.
And the mic is not enough to hit it back there, huh? Okay. When we're talking about curations, we're covering areas including genes and protein structures, gene functions, alleles, sequence variations, phenotypes, nomenclature. We're not just talking about genes and gene models. And Morgan was sharing with us the kinds of things that you can answer with your genome. And so I've listed a few of the things that people usually ask. And you can compare genes across species and across individuals from the same species. But in different settings, in clinical settings, you can look into SNPs. You can look into SNPs and your own data. In a clinical setting, you can compare a patient versus a background population, or you can prioritize variants to try to find candidate genes for disease and for phenotypes. You can make inventories, try to count, do I have all the genes that I suspect I have, look into expansions and contractions of families, looking for evidence of selective pressure across evolutionary distance, and some of you are already working with these things. You can explore genotype to phenotype relationships using the genome. You can characterize the percentage of GC that we have in your genome, assess conservation and uniqueness across phyla, what makes your species, your favorite species, your favorite species. You can infer functional assignments through bioinformatics or through laboratory experiments, and much, much more. Wow, that's very loud. I don't know if this thing has some type of volume, sorry. I could speak louder, but then I would just be screaming. So in order for us to be able to look into these questions and to draw accurate conclusions, we need, I resumed all of this in 3 things. We need good genes, we need good data, and we need to organize our knowledge. And so I'll walk you through the good genes part first. This concept of curation, and I did much like a curator at the laboratory, right? Like looking into literature, published literature, looking into the material, and looking into different things, different avenues of informing a decision. So you are doing the same with your genes. So in the context of genomics, curation takes us to this part of what we have called in the past and introduced to you as the genome train. Imagine this is the life cycle of a genome sequencing project, like the ones that many of you have been involved with. You get your sequence, do your assembly, you take all of this assembly to somebody that runs an automated annotation set of software for you, and then you get gene models that have been predicted. That's where you start this process of manually looking into some of those gene models and then merging a set that contains both the automated annotations as well as the manually annotated ones. And with that, you can run comparative analysis for those genes or gene pathways or gene families of interest. And then once you have been able to get all of your synthesis, all of the results and conclusions out of this, you return this back to the wild for you or for others to run into experimental design and to use it as the beginning of their experimental design template. And this cycle continues. This is not just a one-way highway. And when I speak about manually curating gene annotations, specifically for the work that some of us are already doing here, but many of us have been doing for many years, what we're looking at is specifically looking at those automated predictions and looking at the experimental data that supports them. In this example, I'm simply showing, for instance, this is a gene model that was predicted using one of the automated predictors. It could be Maker in this case, or it could be no one like ours. Yes?
Can you define a gene model?
So there are no such things as genes, only gene models, says Bill Gilbert, who is somebody who has been working for many years on annotation. Basically, a gene model is a representation of the underlying biology, given everything that we know through it, and that we create all of this through algorithms. We call them genes when we give them to you in a browser, and we say, these are all the genes that have been predicted. But truthfully, nobody knows what the ultimate biology looks like. And so instead of saying this gene is the truth, I call it a gene model. And it is up to us to investigate whether or not this is true and to make claims that way.
And it has more specifically green parts, red parts, blue parts?
In this particular example, it's simply the different colors show you the frame for the coding sequence. So this could all be just one color and it would still be a gene model. So in this, I can show you one when I talk about genome browsers, but we can share this now. Imagine that in this case, this is the 5' end. This is the 3' end of the gene. You can't see this very well because this projector is actually blurred, but there's an arrow that tells you that that is the direction of transcription going that way. Each of the thick bars here are exons, and the thin lines are introns, or so the gene model predicted. Right? So the predictor actually said that that was a whole gene model. However, when we went and looked into the supporting evidence data, we have seen gene expression level from aligned RNA-seq reads, and we realized that this is a very high representation of expression levels. There's medium here, and this is just very, very few reads. And so looking at this, we actually realized that there is very, very little to no supporting evidence from the RNA-seq reads telling us that those are actually all connected into a single gene. So these are in fact 3 separate genes and not just one as it was predicted. And it requires us to look into that portion of the genome to be able to make a decision about removing those connecting introns and just splitting that into 3 separate genes. So one question that is very common is like, why would automated gene models be wrong? Isn't that what we all rely on to make our assertions? And the issues are varied, and the best way I can explain it in the very short time that I have is that we have ab initio predictors, which are basically algorithms that are looking into the sequences, and based on the principles of what we know that genes should look like, just on sequence alone, predicting genes. And then we use evidence in order to inform some of those decisions, transcripts, proteins, protein homology. So here is an example from a paper from Daniel Enzi, and the guy at Eccles that writes Maker is escaping me. Wow, I can do it. He's going to kill me. Mark Yandel, sorry. So this gene predictor called SNAP made this as the prediction of the gene model, but adding RNA, mRNA, and EST evidence, this is a whole paper, using an algorithm called Accelerate, it actually predicted a longer transcript. Cross-referencing to other proteins was only able to pick up these little chunks over here because perhaps the sequence similarity was very low in this part. But when we look at the gene annotation that results from synthesizing all of that available evidence, we actually are able to tell that, yeah, indeed, there were 2 exons over here, and this was an alternatively spliced exon, so those are two different isoforms for the same gene, and this was, these, we can have any and all of these representations when we look at our automated annotation, so we need to look a little bit deeper into it and look at it with the evidence in order to be able to resolve them.
This example shows how hard repetitions to produce 5' and 2' ultrasound is an important point, because oftentimes these repetitions completely miss the start side.
Correct.
That's shown on the slide.
Yes. Yeah.
And that's the weakness of the predictions.
Correct.
The strength of the
Correct. Yeah, did everybody hear this? So Claudio is pointing out that this example actually shows how difficult it is to find out, to predict the 5' UTR, the untranslated regions on both ends of the protein. It's actually showing us that very frequently, the automated annotation systems actually make mistakes about this. And Claudio would know, he was teaching us yesterday, again and again, how it was actually necessary to look into these protein domains in order to understand the evolution of those proteins.
There are papers published where you take these predictions in the top and make predictions where the promoters are and talk about regulatory elements. And I think, again, the importance of specific to 5' UTR exons.
Correct.
They're much for the regulatory agency, much for the regulatory.
Imagine if you make a mistake of predicting that 5' UTR here, then you're missing all of this information in the regulatory regions that are upstream from those. So that's very important to highlight. Yes, yeah, that's true. Thank you, Kelvin. So, but this doesn't mean that all comparative genomics is wrong, right? Like of course, there are things that we're capable of doing with the gene sets as they come out of the printer. A good assembly and a good annotation process actually tells us that most genes will be mostly correct. And this is more true for widely conserved genes than for those that are dynamically evolving. And so, so this example, this is Bosco, which is a set for benchmarking universal single copy orthologs. And basically what we're showing with this graph is a number of species. These are all insect species that study past life. So I'm using those examples, but we have some wasps over here and flies and ants. And what it's telling us is that for many of these sets, there is actually a higher, these are all sets that were manually annotated, and that process of manual annotation allowed us to recover a larger portion of the golden set against which we benchmark all of the collections of single copy orthologs. So imagine if you have kind of like that golden set, and you have all of your genes, and you compare them against them, and and the results will tell you your genome had a 70% representation of all of the genes that are expected to show up as kind of like the golden set in the arthropods, or your genome actually was not very, very good and only had a much lower percent of this. So this is just showing that through the process of manual annotation, we were able to rescue much higher numbers in species that have been curated, more like the honeybee and the fruit fly, everybody's friend. Then of course that Bosco set is actually 100%. So why do we want to curate gene models? Do you trust those genes that you're looking in that are coming out of the pipeline? And when you state anything in your publication, are you really sure that there are that many genes, or that you're not missing any, or that you're not overrepresenting any family or members of a pathway? And are you sure that you have the full gene when you're trying to do those molecular evolutionary analysis, or are you only just using half of your gene for those trees? And you want to answer all of these questions because you know that some quality challenged assemblies are going to perform a lot worse than others that have better quality. And really, if you hear nothing else from me this entire week, hear this, and that is for anything that you're going to publish, anything and any statements that you make about a gene, curation helps you reinforce the idea and to reinforce that this is actually true. You're just making sure that what you're saying about this gene is true by looking into that structure. So, of course, in the large scale studies, you're protected. The small scale errors are not that evident, except when they are, and they cause you trouble. So, for the most part, if you're looking into phylogenomics, morphology delineation, the things that that, for instance, Ed and Rebecca do, you do not usually need to go into the specifics of defining everything about your gene, except when you do need it. So many of the most biologically interesting genes and gene families show this highly dynamically evolutionary histories. And this makes manual curation of these genes even more critical. The problem is, for instance, in this family of genes of ionotropic receptors in the mosquito, we were convinced that for the Aedes aegypti, there were just like over 60 or so. And it was because of a master's student sitting down and reviewing the newly assembled genome of Aedes aegypti that they realized they were missing nearly 60 ionotropic receptors, and they had not been visible. So when this happened in the Aedes aegypti, the other mosquito researchers said, we're going to go back and look what's happening in the Anopheles gambia. And let me tell you, these are really well curated genomes that people have been working on for decades, and they had a terrible error. They were actually missing nearly 70 ionotropic receptors that had never been looked at because nobody had ever bothered to go that deep into the family and look at the missing domains. And so now they have full complements of this. So beyond this issue of is the gene there or not, what are the aims of curation? We want to have reliable hypotheses of the putative gene functions that we're asserting for our papers and just in general for our work. And this relies on a confident characterization of the gene evolutionary histories. And this itself, in order to be able to answer that, relies on having a high-quality sequence alignments for robust phylogenetic analysis. And all of this actually hinges on our ability to have complete and accurate gene model annotations. So at the base of answering your questions with certainty is having good gene models to be able to answer those questions. At the Manakin RCN, what does this look like? So we've been wrangling in massaging data for a number of years. It has been, as you all know, a labor of love. We've collected data from a lot of you, and Morgan was just giving us this summary of where all the data are. But there are still things that need to happen, some alignments across species, and we're still organizing transcriptomes in order to be able to display them on our browsers. Another thing that we're doing concurrently, and I'll switch super quick into a spreadsheet here. Is we're actually making, oh wow, that's terrible, this thing. Let me see if I can make it more visible. If not, I can just tell you what it says. So this is a spreadsheet that is online right now that we have sent around to the Manakin RCN. It's a place to collect your genes of interest, your pathways of interest, your families of interest, and it collects information such as the name that you know, the common name, any symbols that you have for each of these genes or for the gene families, and tries to identify them both in the Manacus, I think you say and Bitraficada. So you're getting information from the assembly version, from the scaffold that it is, where it is located, the coordinates in that. But in addition to getting those identifier information, you're also getting information about the particular gene family or pathway where it is, where it participates, a member of. We're trying to get information as well about orthologs from other species and which species those are, any information that we can get about the genome function. And if you have gene ontology term for that function, we're also collecting that, as well as any supporting evidence, such as evidence codes, if you are familiar with those, if not, you have to write it, and then publications that are actually supporting that work as well, and then any other comments that you want to give us about the gene of preference. So if you haven't already visited this spreadsheet, then please visit this spreadsheet. So what is the purpose of this? What is going to happen next is that you are going to become the curator of that gene or gene family, or you're going to enslave a graduate student to become the curator of that gene or gene family. So whatever your interests are, our hope is that you're actually going to be able to get the finalized gene models from this. We have been setting up a curation software that is almost done. We're waiting on some data to be updated, and we are hoping to do the training and curation exercise for the This would be the second time, with all of you. Hopefully we're going to be able to do this together in a room in November. And if this doesn't happen together in a room in November, it will happen online, and we will do this via Zoom. I've actually successfully done this many times. The last time was 125 researchers across 3 different time zones in Australia, and we were together for 3 hours. I'm hoping that hours can be longer. Actually, if we can invest a full day or 2, would be even better. If we cannot co-locate, we will do it virtually. And then one important aspect of this curation process will be that not only will we be training ourselves in order to become familiar with the software and with the curation process as a whole, but also you will become the trainers of your own teams. And I warn you, it becomes addictive. You may like it a little too much. And then we'll do the collection and analysis so that we can discuss results together. This helps you at different levels, just in the absence or presence or presentation of each of the gene families that you're interested in, but it also helps you identify things at the level of new domains or diversification or an expansion or contraction of the family on the species that you're working in. Just to give you, this is the only details that I'm going to give you about the process of gene curation, but this is to get you in the mindset of what you would experience. Many of you have actually done curation in Apollo before. We have done these exercises together. You basically look for a region of interest or a scaffold or a gene that you already know where it's located. And you look through the evidence that we have. When I talk about evidence, I talk about all of the experimental data that has been collected and that is available for all of these, for each of the species that we work with. And so you look at RNA-seq data, or you look at any other type of data in support of these genes. And you determine whether or not one of those tracks of experimental data is going to be, or the gene model that has been predicted is going to offer you a starting point for editing. This is in the abstract for now, but when we talk about browsers with Sammy in a second, it'll become clearer. You make any modifications to that gene model that are necessary. I have been actually looking into the gene models that were predicted for Migraphilic taura, and I have been happily encountering many instances in which the gene that one of you proposed was actually predicted accurately, and the transcriptomic data that is already there is actually very good support for those predictions. It actually, the normal pipeline from NCBI managed to predict at least 3 or 4 isoforms for some of those that have differential splicing. So maybe this will be a very rewarding exercise, because we will be able to see from the 10, 20, or 40 genes of interest that we're interested in, there are only going to be some modifications. So that's good news. And for the other genome, for the majority of the genomes, the gene prediction would be, of course, expected to be fantastic and easier to go through. Once you make those modifications, then you comment and then report and finish. And the last thing I want to say is just an introduction to the data management team, basically. So in addition to having good genes to make good assertions and get good conclusions, you also need to have good data. All of the data that we produce, not just the genomic data, but this is true of all of the data that you and I produce together. Our data must be infinitely shareable. Its reuse increases its value. The more data are reused, the more data they generate. And think about this as well, because data are perishable. It's okay to archive when it is no longer serving the purpose of informing what you're trying to assert. Combining and integrating data sets increases the value of the individual data sets. So it's important that we think in the context of connectedness, interconnectedness of the data, because having all of this incredible wealth of information in isolation is not very helpful, but that's why a research coordination network tries to bring everything together into a single space. And the more accurate the information is, the more useful it is. And remember that not always more data is better. Many of you have probably heard this. Who has heard of FAIR before? Okay, so FAIR is an acronym that was born a couple of years ago, and it tried to describe the things that we need in order to keep our data useful and in order to make our data meaningful. So data should be fair, findable, accessible, interoperable and reusable, basically, meaning that it should go into a place that is publicly accessible, or at the very least that can be found and then you can easily log in. You should serve your data, so it should have good identifiers in order to be able to be findable and accessible. It should be connected, like I was saying, connected data is more powerful, and you should be able to reuse this data. But it's very hard to get all the way to FAIR. So we actually think that in order to get from findable and accessible to the entire FAIR, you also need a little TLC. You need to make your data traceable. You have to be able to give attribution to the generators of the data. Thank you. You have to be able to give attribution to the generators of the data, as well as credit to those that are working with your data. I don't know if you guys think about this often, but I would like for you to start thinking about it if you don't already. Think about how you're licensing your data, right? We're a research coordination network. We are sponsored by the National Science Foundation of the United States, which demands that all of our data has to be out there publicly available without any embargoes, right? And once we generate this data that go to the NCBI, for instance, if the NCBI is going to run our genomes for annotation, they have to put it out there immediately. And all of those things have to go with some type of license. And I don't know that we have sat down and thought about how our resources are licensed. So this is an important conversation, and I think that management team will have more to say about licensing. And then the connected part is about giving all of our data appropriate identifiers and making sure that we can trace everything that is happening with our data, and that we don't have wacko identifiers that are either cryptic or that are redundant and that are going to end up making it confusing for us to understand it. That's why in that spreadsheet that I showed you earlier, the first thing you see on the left is 6 columns of trying to understand the identifiers that we're using, what you're using from the NCBI identifiers, as well as the not just where they are located, but also the individual identifiers for each of the genes. Anyway, so that's oh, and these are the 3 things. That's how it's explained. All of these slides are going to be made available. Betty's actually going through the slides and making a recording, and all my slides are shared with a CC by 4 license, which means you can all use them any way you want, as long as you please take attribution or describe where they came from. I'll provide them on PDX so that you can reuse them however you want them with attribution. And with that, I think I hand it over to the data management team.